Okay. Hello everyone. Is my voice audible to you all? Can everyone confirm? Yes. Am I audible to you all? Good evening everyone. Please confirm if I'm audible or not. Okay. Hello everyone, welcome to Botany Insider. Good evening Shivani. Good evening Bhargavi. Let's wait for other students to join and then we'll start our class, okay? Is there any disturbance in my voice? Yes, please confirm. Okay everyone, we'll start the class now. There are 16 students in the class. Hello everyone to you all. So this is our first class for unit number 13. And unit number 13 is method in biology. Okay. This is my first class with you all. Hello everyone, my name is Kushi Jain and I am your new educator at Botany Insider. Okay, so first of all, we have two courses running. First is full course and other one is crash course. If you want to prepare for the December 2023 exam, then you can enroll in these two courses because these two courses are designed. Okay, Bhargavi, little disturbance. So I was saying that these two courses are designed in a very structured manner and they will surely help you with your preparation. Also, we have a notes booklet, so you can purchase this through our website and from our app. You can download our app from Play Store and iOS as well. Okay. Other than this, we'll be launching soon with a new course which is our PYQ course. So this is going to help you with your preparation and your doubt clearance regarding all the PYQs that were there in CSIR NET exam. Okay, so you can surely enroll. You can surely enroll in this batch. Okay, this will come soon. So first of all, we are starting with all the techniques and their application. See, with the Unit 13, there is a thing that CSIR usually asks match the following questions and statement-wise questions. And in match the following question, the majority of the questions were from techniques and their application. Okay, and these are the easiest question to target because these are the questions that you can attempt with the most accuracy possible. You can even get 100% accuracy in these questions. voice not clear. Bear with me for few seconds and you will get my voice clear. Okay, Mukesh? First, focus on the class and uh, understand the topic. Okay? So we are dealing with all the techniques and their application that has been asked in CSIR. Good evening, Nisha. Good evening. Is my voice better now? Yes, please check. Is my voice better? Yes, please confirm. Is my voice better? Good evening, Lina. Good evening. So, as I was telling that we are dealing with unit number 13 and all the techniques it contains and their application. Okay. So, majority of the questions that are asked in CSIR are match the following based questions. Right. So, for match the following questions, you need to learn all the techniques and their application part only. You need not to go into the detail of the techniques for the first match the following type of questions, right? So in these questions, I have tabulated all the techniques and their applications. All the techniques that have been asked in CSIR before, I have majority of the techniques listed here. So the first technique is cloning vector. Okay. In cloning vector, what we use it for? We use it for foreign gene transfer. Let me change the. So we are dealing with cloning vector and the foreign gene transfer. So in foreign gene transfer, what we do is that we have a gene of our desired interest that we want to clone. So we use the cloning vectors to integrate these genes into the organism. Okay, and then we can use these genes and analyze the functions of the gene and their expression into the organism using cloning vectors. Other thing is gene knockout and gene knock-in techniques. For gene knockout and gene knock-in, we have three types of technique. First is neo-resistance and TK gene. Second is CRISPR-Cas technique. You must have heard of this technique because this is a very popular gene knockout technique and this has been asked in CSIR multiple times in the match the following question. Okay, and then we have pre-lock system. Okay, pre-lock T system. So these are the three techniques. Don't worry, we'll be dealing with these techniques in the detail in coming lecture also. But for here, for the application part, you just need to know that these are the three techniques that we use for gene knockout and gene knockout. Then we have PCR, which is polymerase chain reaction. In polymerase chain reaction, we use this technique for protein and DNA amplification. Okay, and the other technique is RACE. RACE stands for rapid amplification of cDNA ends. Okay, what we do in this technique is that we have an mRNA. Okay, and we all know that this mRNA have 5' and 3' end. In the 3' we have a poly A tail, and in the 5 prime end we have a cap. So whatever gene that is present in the ends of mRNA or cDNA, we determine these genes using the technique known as RACE, because we all know that 5 prime cap is the most important site for ribosome binding site. Okay, so for this purpose we need to know the sequence of the gene that is present, or what type of genes that is present here. So we use the technique RACE, which is rapid amplification of cDNA ends. Okay. Then we have three most important molecular markers. We'll be dealing with in detail all about these markers in our coming lectures. But for here, we just need to know that these are the molecular marker and they have a specific function. The first one is RFLP, then we have RAPD, and then we have AFLP. RFLP is restriction fragment length polymorphism. Then we have RAPD and AFLP. The RFLP is of codominant type and RAPD is of dominant type. We use RFLP for parental and criminal analysis. Okay, and RAPD is used for evolutionary studies when we want to differentiate between different phylogenetic trees. Okay, when we want to differentiate between different organisms or different species, then we use RAPD. And in case of AFLP, we use it for parental and pedigree analysis in DNA fingerprinting technique. Okay, then we have UV visible spectroscopy. It is used for concentration determinant. Then we have three techniques for protein structure determination. First is circular dichroism, then we have NMR, and then we have X-ray. First thing first, that you want to remember that what type of structure it determines. For circular dichroism, we use it for secondary structure determination. And for NMR and X-ray, we use it for tertiary structure determination. Now how you all can remember it, that circular dichroism have two letters. So we can remember that it is used for secondary structure prediction. And NMR has three letters. So we can use it for tertiary structure prediction. And for X-ray, it is used additionally for DNA structure prediction as well. Okay. Then we have agrogel electrophoresis, which is the most common technique used in almost all the labs, majority in molecular biology labs, and it is for DNA molecular weight determination. Then we have transcriptome analysis. It is also for DNA molecule weight determination. The other three are type of spectroscopies. First is mass, then we have infrared, and the last one is fluorescence spectroscopy. In case of mass spectroscopy, we use it to analyze the mass of biomolecule or to identify the molecule mass. And then we have infrared spectroscopy. It is used for identification of functional groups. What kind of functional groups are present in that particular biomolecule? We use IR spectrometry. Then we have fluorescence spectrometry, and we use organic group determination for it. Okay, we use fluorescence spectrometry for organic group determination. I hope this table is clear to you so far. Please confirm. We have 20 students in the class. Yes. If all the techniques and their application is clear to you or not. Good evening. Okay. Now we have other important techniques as well. First is hybridoma technology. It is the most important technology of immunology in which we use it to produce monoclonal antibodies. Okay. We use hybridoma technology to produce monoclonal antibody. The other two important immunology experiments are radioimmunoassay. RIA stands for radioimmunoassay, and the other one is ELISA, which stands for enzyme linked immunosorbate assay. Okay. RIA and ELISA both are used for quantitative and qualitative analysis of the protein, and rather we say that it is used for antigen-antibody interaction. We can qualitatively and quantitatively analyze the presence of antigen and antibody interaction. Then the other techniques of cell biology are flow cytometry and FACS. FACS stands for fluorescence activated cell sorting, assisted or activated cell sorting. Okay. In case of flow cytometry, we differentiate cell on the basis of their morphology and function, and in case of fluorescence activated cell sorting, we use it for cell sorting and for sorting the apoptotic cells. Apoptotic cells are cells that have the signal for self degradation. Okay. And what was the signal? The signal is known as eat-me signal. Can you all tell me what is the name of the phospholipid which is used in eat-me signal or the apoptotic signal? Yes, tell me the name of the phospholipid which is used in the apoptotic signal. The phospholipid which is, yes, very good. It is phosphatidylserine. Then we have different kinds of blotting experiment. For DNA analysis, we have southern blotting. For mRNA analysis, we have northern blotting. For protein analysis, we have western blotting. And for post-translation modification, we have eastern blotting. So it is very important to remember the type of blotting and for which biomolecule we are using it for, because it has been asked in CSIR multiple times. I'll be showing you a question in which these blottings were asked. Okay. So southern blotting is used for DNA analysis, northern is for mRNA, western is for protein, and eastern is for post-translation modification. Then we have DNA microarray, which is used for many gene expression analysis. The other technique is FISH and FRET. So FISH is used for in vivo DNA and gene analysis, and FRET is used for protein-protein interaction and nucleotide annealing. FISH stands for fluorescence in situ hybridization. In situ hybridization. And FRET stands for fluorescence resonance energy transfer. First is used for in vivo DNA expression analysis. FRET is used for protein-protein interaction and nucleotide annealing. And fast display is also the technique which is used to analyze the protein-protein interactions. Then we have surface plasmon resonance, which is to analyze the interaction of biomolecules, and different type of chromatography, which is used for purification of protein and RNA. And a specific type of chromatography is affinity chromatography, which is used for mRNA purification. Okay? Is everything clear this far? If anyone have any doubt, you can ask. Please confirm if everything is clear in this table. Okay. Then we have another technique which is also used for protein-protein interaction. The first was FRET. Then we have fast display. Lina, we will discuss all these techniques in detail in the coming lectures also. So don't worry about the difference, just understand the application part only. Okay? There is a big difference between FRAP and flow cytometry. In flow cytometry, we separate cell on the basis of their structure and function. Okay? But in case of FRET, which is fluorescence recovery after photobleaching. So in this technique, what we do is we check the diffusion of the lipids, the lateral diffusion. We study the lateral diffusion of the lipids. Okay. Don't worry about these techniques. We'll see these techniques in detail. Okay. Then we have five major types of microscopy. In case of light microscopy, we use it to analyze the 2D image of the cell and of larger organelles. In case of confocal microscopy, we use it for 3D image of cell and tissue. In case of phase contrast microscopy, we use it for living natural cell, and this has been asked in CSIR. Okay. Then we have two other technique, which is SEM, surface electron microscope, and transmission electron microscope. In case of surface electron microscope, we use to view the surface of the cell only. And in case of transmission, we use it to view the internal part of the cell and the organelles. Okay. This was all about the techniques and their application. Now we will see questions that were asked in CSR. So confirm if everything is clear, then we'll solve the questions together. Okay. If everything is clear, then solve this question. This has been asked in CSIR in the year 2019. Read the question carefully and then mark the answer. this is the most basic type of question that is asked in CSIR Part C. So it is a 4-marker question. Read the question carefully. There are 29 students in the class and I want all of you to answer the question. Yes, very good. Answer everyone. Yes, the answer is option number 2. Just by elimination method, you can solve this question without even knowing all the techniques. Okay. I have already told you about RACE, which is the rapid amplification of cDNA ends, and here it is written determining the ends of mRNA. So A gets matched with third. Then we have south-western blotting. For southern we have DNA and for western we have protein. So it is for DNA and protein interaction. So B gets marked with 2. So if we mark A with third, with just one technique we can answer the question correctly. Okay, we don't even need to know all the other 3 techniques. Just with one technique we can answer the question. So you can see that CSIR is asking very easy questions. Now answer this one. This has been asked in CSIR 2022. So it is a very recent year question. Read the question very carefully and then mark the answer. If you can't get to the answer, just try to attempt it. Even if you get this question wrong, no worries, but it is a very easy question. Quickly mark the answer. We have 27 students in the class and only 7 of you mark the answer. Yes, the answer is option number 2. See, you need not to even match the answers. The CSIR already given you the correct answer with its match only. For three-dimensional structure, we use NMR. For ionic charge, we use isoelectric focusing. In affinity chromatography, we check the binding specificity. And for molecular size, we need ultracentrifugation. So the correct option is option number 2. Now this is the last question for today. It came in 2017 and it is again a very easy question. So mark the answer, read the question carefully. Quickly mark the answer. Yes, the answer is option number first. None of you got the answer correct. No worries. But for race, we know that it is used for the identification of 5' and 3' end of the transcript, mRNA transcript. Okay, so D gets marked with 2. So we eliminate the option number 2 and 4. And for ELISA, we know it is the quantification of transgene expression. So C gets marked with 4. Option number first is correct. Okay. This was all for today. I hope the lecture was clear to you all. If you have any doubt, you can ask. Was the lecture helpful? Okay. Now we have 2 courses running. Again, I am reminding you all that we have full course running and a crash course running. You can enroll in any of the course. Also, we have a notes booklet for the revision and better preparation of your exams. So you can get the notes booklet as well. And we'll be launching a PYQ course for the session of December 2023. So make sure to enroll in the batch. Okay. Thank you so much for your participation and presence in your class. Yes, and be there in the class 8 p.m. tomorrow. We are running this marathon on the daily basis on YouTube for free. Thank you so much. Thank you, everyone. Okay, Mukesh will provide the PDF on our Telegram channel. So you can follow us on Telegram channel as well. The link will be provided in the description box. Bye bye.